Welcome back to the Deep Dive. Today we're looking at something really, well, monumental. A shift in personalized medicine.
Yeah, it's this convergence point, isn't it, between two massive technologies we track: synthetic biology and AI.
Exactly. And we're not just talking small steps here. We're looking at the groundwork, maybe the theoretical foundation, for a completely new kind of medical system.
That's right. If you follow either field, you see the power. But putting them together, that unlocks this idea our sources talk about: parallel simulation.
Yeah, well, simulation. Okay.
So our deep dive today is really mapping out what you'd need for that future. A future where you could essentially digitally clone a patient-specific biology.
Like a digital twin.
Precisely, a digital twin. And at the same time, design and test a super customized bioengineered treatment against that exact twin.
Wow. Okay, so testing the drug on the virtual patient before it ever touches the real person. That's the goal.
That's the ultimate shortcut, yeah. And our sources, they detail some real breakthroughs already happening: advanced AI reasoning, virtual patient stuff, rapid molecular design. It makes the whole thing feel much more viable.
Right. And you, listening, you came here for that quick, deep understanding. So let's jump right in. The engine driving all this has to be the AI, right? The advanced AI. Okay, so if we're serious about simulating the human body,
Yeah.
I mean, it's chaos in there, right? Billions of things happening at once: metabolism, signals, immune response.
It's incredibly complex, yeah. Nonlinear doesn't even begin to cover it.
So we need an AI that's more than just a data cruncher.
Yeah.
It needs to actually reason like biology does,
Yeah.
almost. The sources keep mentioning deep think.
Exactly, deep think.
Wait.
Because simulating that biological complexity, it demands flexible, rigorous, and crucially fast reasoning. Think about it.
Okay.
Your standard step-by-step computation, it just buckles under the weight of all those interacting variables and, say, a disease state.
Right.
If you want to accurately model how some new protein interacts with everything else already going on in a cell, well, the AI needs to explore countless possibilities at the same time.
And we actually have proof now that AI is hitting that level, that kind of flexible reasoning.
We do. And the evidence comes from a pretty surprising place.
Which is?
The International Mathematical Olympiad, the IMO.
The math competition for high schoolers. The really, really hard one.
That's the one. The most prestigious competition for pre-university math whizzes. It tests deep abstract thinking: algebra, geometry, combinatorics, number theory, stuff like that.
Seems a long way from biology. Why is that relevant?
Because it's about the type of logic needed, that rigorous, creative problem solving. It's profoundly relevant for untangling biological systems, too.
Okay, so what happened at the IMO?
The breakthrough was in the 2025 competition. They used an advanced version of Gemini DeepThink.
Gemini. Okay.
And it didn't just, participate. It officially hit gold medal level performance, scored 35 out of 42 points, which means it solved 5 out of the 6 problems perfectly. And these problems are notoriously difficult.
35 points. That's better than most of the human competitors, right? These are prodigies.
Better than about 95% of them, yeah. But for our deep dive, for the medicine angle, the big deal isn't just the score. It's how it did it and how fast.
Ah, okay. Tell me about that.
Well, let's contrast it with the previous year, IMO 2024. You had systems like Alpha Geometry, AlphaProof. They did well too.
Right.
But those systems needed human experts to spend ages translating the problems first, taking the normal text questions and putting them into these very specialized, formal computer languages like Lean.
Okay, so a human translator step.
A massive one. And even after all that translation work, getting the actual proofs, it took 2 to 3 days of computation.
Days. Yeah, that's completely useless for trying to simulate biology in real time.
Precisely. Just impractical. But this year, the advanced Gemini DeepThink, it just smashed those limits. It worked end-to-end entirely in natural language. It took the official problem text, just like the students get, and generated the rigorous proofs directly. No human translation needed.
Okay, that's huge. And the speed.
That's the kicker.
Yeah.
It completed the whole set of problems within the official 4.5-hour competition time limit.
4.5 hours instead of days.
Exactly. That leap in speed and the flexibility working without needing that human translation bottleneck that's absolutely non-negotiable if you want to take this tech from abstract math and pivot it into the messy, time-sensitive world of life sciences.
So what's the secret sauce there? It sounds like it's not just faster chips. The sources mention something called deep think mode.
Yeah, deep think mode. It's basically an enhanced way of reasoning. It brings in parallel thinking.
Parallel thinking.
Yeah, think of it like moving beyond the old way, the single step-by-step logical chain, A leads to B leads to C. When deep think mode kicks in, the model explores lots of different possible solutions or pieces of a proof all at the same time. It tries combining them in different ways before it settles on the final answer.
So it's like running multiple simulations inside its own head simultaneously.
That's a good way to put it. Running many complex, abstract simulations in parallel internally.
And that structure, that parallel exploration, that sounds exactly like what you'd need for biology, where everything is happening at once.
Absolutely. You're not just tracking one gene, right? You're tracking hundreds, thousands of things interacting constantly.
Metabolism, proteins folding, genes turning on and off.
All interconnected, all in feedback loops. Modeling the human body is fundamentally a parallel processing challenge.
So the fact that an AI can now handle multiple complex mathematical variables at the same time, like it showed at the IMO, that suggests it has the chops for biological simulation too.
It strongly suggests we have the brain needed for this parallel simulation idea. The engine is powerful enough.
In DeepMind, they're not just leaving this in the realm of math competitions, are they? They're pointing it at biology.
Oh, absolutely. The sources confirm this. They list projects we already know about, like AlphaFold for protein structures.
Right, AlphaFold.
But also AlphaGenome for looking at genomes, AlphaMissense for figuring out what mutations mean, AlphaProteo for complex protein machines. They are clearly aiming this advanced reasoning power right at the core components of life.
Okay, so connecting the dots. The IMO result confirms the computational backbone. It can handle the complexity.
Yeah, the complexity of the body and also the complexity of engineering treatments. Which leads us to the next piece of the puzzle.
Which is building the body itself. The simulated patient.
Exactly. The highly accurate, flexible, simulated patient.
Okay, moving to the patient side. Now, in medical training and sometimes drug testing, they've used human actors, right? Simulated patients or SPs?
Yeah, for a long time. But those traditional SPs have some real limitations, especially when you think about the scale needed for this parallel, personalized medicine idea. Well, the sources point out a few big ones. First, cost. Training and hiring good actors or medical pros for these roles, it gets expensive.
Makes sense.
Then there's just the workload. And maybe most importantly, consistency. Can a human actor perfectly portray the exact same subtle symptoms and responses every single time, all day long?
Probably not perfectly. They might get tired.
Yeah,
Very slightly.
exactly. And that tiny variation could compromise the standardization you absolutely need if you're evaluating a student rigorously or testing a drug reliably.
Right. Consistency and scalability. Those seem like the big hurdles for human SPs. You can't run a standardized trial if your patient is a bit different each time.
Which brings us to the virtual simulated patient, the VSP.
Okay. Using AI.
Using large language models, LLMs, their rapid development, especially how good they've gotten at conversation, at role-playing, it makes them a really feasible alternative, scalable, and potentially highly consistent.
But it can't just be a chatbot pretending to have a cough, right?
Yeah.
For it to be useful medically, for training, or this simulation idea, it needs more structure.
Absolutely. It needs to be more than just conversational. It needs clinical utility. And the sources talk about a specific framework for this called CureFun.
CureFun. Okay. What does that do?
It's described as an integrated model-agnostic framework, meaning it can work with different LLMs underneath. And it has two key functions.
All right. Function one.
Function one is pretty straightforward, natural conversation. It basically enables a realistic chat between the trainee or maybe a diagnostic AI in the future and the simulated patient. Captures the nuances of a real consultation.
Okay, that makes sense.
Yeah.
Dialogue. What's function two?
Function two is, I think, the really interesting part for our parallel simulation concept, evaluation.
Evaluation of the patient.
No, evaluation of the student or the AI interviewing the patient. CureFun has this sophisticated mechanism built in to assess the clinical quality of the interviewer's dialogue flow.
Ah, so it judges how good the doctor's questions are.
Essentially, yes. It can give feedback, either in real time or afterwards, on how to improve clinical inquiry skills. Are they asking the right questions, following up appropriately?
Wow. That ability to evaluate the interaction based on clinical standards,
Yeah.
that makes the VSP much more than just a puppet. It proves it can simulate professionally.
Right. It shows proficiency in simulating these scenarios reliably. The sources say this VSP approach, using CureFun, leads to more authentic and professional dialogue flows compared to just using a standard chatbot.
So it's consistent, it's scalable, and it can even evaluate the interaction quality.
And that evaluation power, that consistency,
Yeah.
it led to this really unexpected, almost revolutionary second use case for the VSP.
Oh, What's that?
Think about it. If the VSP is really good at acting like a patient and evaluating the person interviewing it, what happens if the interviewer is another AI, another LLM?
Wait, you use the virtual patient as a standardized test
Yeah.
to grade a virtual doctor AI.
Exactly. Researchers are using CureFund's evaluation capability to assess the performance of different medical LLMs.
That's brilliant.
They use the VSP as this consistent, high-fidelity benchmark patient. It lets them measure the diagnostic skills of various AI doctor models.
Yeah. So it gives us insight into how good these LMs actually are at diagnosis, their strengths, their weaknesses.
Precisely. It helps figure out the potential and the limitations of using LLMs as virtual doctors, and that completes the patient-side piece of our simulation puzzle.
Okay, this really clicks now. The VSP isn't just a placeholder. It's a crucial standardization tool.
Yeah.
For parallel simulation to work, the AI designing the treatment needs the right input from the diagnostic AI, the virtual doctor.
Correct. Garbage in, garbage out.
So having this consistent, scalable VSP that can evaluate the diagnostic AI, it adds a layer of rigor you just couldn't get before.
It ensures the patient model we're simulating against is consistent and clinically accurate, no matter how complex the scenario. That consistency is the bedrock. It's what lets us reliably test the treatments we're about to design.
Okay, so we've got the brain, the deep think AI doing the heavy parallel reasoning. We've got the consistent patient, the VSP using CureFund. Now we need the factory. The way to rapidly design brand new personalized treatments.
And that's where synthetic biology, or SynBio, comes in, turbocharged by AI.
SynBio. This field has been growing fast, hasn't it?
Explosively fast. The scale is huge now. The source is, quote, the global market value. It was $6.8 billion back in 2020.
Okay.
The projection for next year, $19.8 billion.
Wow, nearly triple in just 5 years. What's driving that?
It's the push for sustainable, efficient production systems, not just for health, but for industry, agriculture, the environment. SynBio is trying to apply engineering principles like modularity, standardization to biology itself. Make it predictable, make it designable.
And when we talk personalized treatments, we're often talking about designing specific molecules, right? Like proteins or enzymes.
Yeah, exactly. The functional workhorses of the cell,
Right.
the molecular blueprints.
Yeah.
And AI is drastically speeding up how we design these at the most fundamental atomic level.
How do we used to do it before AI really jumped in?
Well, the sources lay out two main traditional approaches. First, rational design.
Rational design. Sounds logical.
It is. It relies on having detailed information already, the protein sequence, its 3D shape, how it works. Scientists use that knowledge to make very specific, targeted changes, often helped by computer simulations like molecular dynamics.
Okay, so knowledge-driven. What's the other way?
The other way is more random, in a sense. It's directed evolution.
Like Darwin in a test tube.
Pretty much, yeah.
Yeah.
You mimic natural selection. You introduce random mutations into the gene, maybe using something like error-prone PCR, create a library of variants, and then screen them to find ones with improved function.
Effective, I guess, but sounds slow and maybe hit or miss.
It can be very effective, but often time-consuming and needs really good high-throughput screening methods.
So early AI tools like AlphaFold, they were meant to boost the rational design side, right?
Yeah.
By predicting the 3D structures.
That was the promise, and AlphaFold was a massive leap forward in predicting structures with high accuracy. But there's a key limitation when it comes to designing something completely new. De novo design.
What's the limitation?
AlphaFold works best when the protein sequence you're asking about is similar to structures we already know from experiments. Its accuracy relies heavily on that existing data.
Ah, so if you need a protein unlike anything seen before, maybe to target a unique marker on a simulated patient's cancer cell, AlphaFold might struggle.
According to the sources, yeah. Its performance for predicting completely novel structures, the kind needed for truly personalized medicine tailored to a VSP simulation, it falls short of what's really needed.
Okay, so if the goal is true invention, creating something brand new from scratch to solve a unique problem, we need more than just prediction. We need generation.
Exactly. We need generative power. And that's where the next wave of AI models comes in.
Like what?
Well, one area is ML-directed evolution, MLDE, machine learning-directed evolution.
Okay, using ML to make the evolution process smarter.
Basically, yeah. Instead of just random mutations and brute force screening, ML algorithms guide the selection process intelligently. They learn the relationship between sequence changes and function much faster.
And that speeds things up.
Dramatically. The sources mention an example where MLDE found high fitness protein variants much more efficiently. It reduced the number of needed experiments, the training set size, by 81 times compared to a simpler optimization method.
81 times faster to get to the good stuff. That's huge.
It is. That kind of speed up changes the game.
But for pure de novo design, starting from just specs provided by DeepThink, maybe, and creating a functional proteins is a tool for that.
Yes. The sources point to a really exciting novel generative model called RF diffusion. That stands for Rosa-Fold diffusion.
RF diffusion. Okay, diffusion models are big in image generation.
Yeah.
How does it work for proteins?
It's based on similar principles. In simple terms, it learns to reverse a process of adding noise or randomness to a structure. It starts with a kind of random, noisy molecular arrangement,
All right.
and then step by step it refines it, removes the noise, until it arrives at a stable, plausible, and hopefully functional 3D protein structure that actually meets the design criteria you gave it.
So you tell it, I need a protein that binds tightly to this specific target from the VSP, and it kind of works backward from randomness to generate the molecular shape that does that.
That's the core idea. It combines powerful structure prediction networks like Rosa-Fold with these generative diffusion models. The sources really hype this up as potentially revolutionizing de novo protein design.
So in our parallel simulation system, DeepThink figures out the strategy. RF diffusion designs the custom molecular tool needed for the VSP.
Exactly. It's the custom molecular design engine, guided by the AI brain, creating the treatment to test against the virtual patient. Fast.
Okay, designing it on a computer is one thing, but getting it ready for actual production, even small scale, that's another step.
Yeah.
How does the AI SynBio toolkit help bridge that gap?
It comes back to those engineering principles, modularity and standardization.
Modularity first, making things like interchangeable parts.
Yeah, treating biological functions like Lego bricks you can snap together. This is key for making made-to-order molecules quickly and commercially viable. And AI tools like deep docking massively accelerate this part.
Deep docking, what's that do?
Traditionally, scientists would test maybe one design idea at a time computationally. Deep docking can generate and evaluate millions of potential solutions for how molecules fit together, how drugs might bind to targets, all simultaneously.
Millions at once.
Yes, it brings that sheer computational speed to bear on the complexity of molecular interactions. You can optimize a potential drug candidate in, well, maybe minutes or hours. Instead of weeks or months or years, it just changes the economics.
That's incredible. Okay, that's modularity sped up. What about standardization? Moving from a one-off discovery to something reliable you can make again and again.
Standardization is absolutely critical for industrial scale. The sources mention tools like NP classifier and dark MPS.
NP classifier and dark MPS. Catchy names.
What they do is pretty cool. They're designed to analyze novel chemical compounds and actually predict the likely biochemical pathways needed to synthesize them, even for unnatural or highly modified molecules.
So they figure out the recipe automatically.
They help predict it, potentially bypassing a lot of the slow, painstaking lab work usually needed to figure out how to actually make a newly designed molecule. This speeds up the transition from a digital design to a standardized biological part ready for someone to produce.
Okay, so AI designs the molecule. AI helps figure out how to make it. Then the final step is actually making it, moving from the simulation to the real world, or at least a real bioreactor.
Right, and this is where another type of simulation comes in. Digital twins, but this time of the manufacturing process.
A digital twin of the bioreactor.
Exactly. Integrated mathematical models that simulate what's happening inside the tank. They aim to predict things like productivity levels, yield, and help you rapidly figure out the best conditions, temperature, pH, nutrient feed rates, whatever.
So you optimize the manufacturing virtually before you waste time and materials doing it for real.
Precisely. The sources mention tools like the Automated Recommendation Tool, or ART. ART can look at potential microbial strains, predict how productive they'll be under certain conditions, and streamline the whole scale-up process. Makes it predictable, efficient, cost-effective.
Okay, the whole chain is coming together. DeepThink reasons, VSP models the patient RF diffusion deep docking, design the treatment, standardization tools, plan the synthesis, digital twins, optimize the production.
That's the synergistic loop. It's all connected.
Now, this focus on personalized medicine is obviously huge, revolutionary, but the sources seem to suggest this AI and SynBio combination. It's bigger than just human health. It's foundational for a whole bio-based economy.
That's absolutely right. The tech isn't staying confined to medicine. It's rapidly being applied elsewhere, like optimizing production in plant systems.
Lance, how so?
Well, Think about using plant cell cultures, or things called hairy roots, to produce valuable stuff, maybe pharmaceuticals, specialty chemicals. It's attractive, but really complex to get right,
Why complex?
because you have to optimize so many things at once. The plant's genetics, the genotype, the soup it's growing in, the culture media, the growth regulators you add, they all interact in complicated ways.
Sounds like another job for AI then. Lots of interacting variables.
Exactly. The same kind of AI and machine learning we've been talking about. Artificial neural networks, ANNs, genetic algorithms, GAs, multi-layer perceptrons, MLPs. They're being used to map these complex relationships.
So the AI learns how tweaking the nutrient mix affects the yield of the chemical you want.
Precisely. It can model those nonlinear interactions that are hard for humans to figure out through trial and error.
Is there a concrete example of this?
Yeah, the sources detail a case study using MLP combined with genetic algorithms. They used it to predict and presumably optimize the production of paclitaxel.
Paclitaxel, that's
Yeah.
a major anti-cancer drug, right?
It is. High value. And they were predicting its production in cell cultures of Corylus avellana, that's the hazel tree. The GA helps find the best input combinations. The MLP models the resulting yield.
So you could potentially grow complex drugs in optimized plant cell factories instead of just traditional chemical synthesis.
That's the idea. It opens up new avenues for biomanufacturing.
Okay, so individual plant systems. What else? The sources mentioned plant microbiome engineering. Sounds important for farming.
Hugely important. This could be a game changer for sustainable agriculture, potentially reducing our reliance on chemical fertilizers and pesticides.
How does it work? Engineering the microbes that live with the plants.
That's the core concept. Synthetic biologists are working on developing smart microbiomes. These are communities of engineered microbes designed to live with the plant on its roots or leaves.
And they do helpful things.
That's the goal. They could be engineered to sense stress like drought or high heat and then actively respond. Maybe they release protective compounds or help the plant grab more nutrients, ultimately boosting crop yield and resilience.
Wow, like a living defense system or support system for the crop. Sounds amazing for sustainability. But the complexity must be enormous. You're dealing with a whole community of microbes interacting with a plant.
And the data generated is just gargantuan. That's the critical challenge here.
What kind of data?
Multiomics data. You need to understand everything happening, the metabolomics, what small molecules are around, the metatranscriptomics, which genes are active across the whole microbial community. It's vast, complex data sets, far too much to handle manually.
So you need serious data infrastructure just to make sense of it all.
Yeah.
You can't simulate or engineer a microbiome without that.
Absolutely not. And the sources highlight this need, pointing to things like the National Microbiome Data Collaborative, NMDC.
NMDC, what's that?
It's a data portal, a big one. The source says it supports 10.2 terabytes of multiomics data just on microbiomes.
Terabytes.
And its main job is standardization, making sure all this data from different studies can be found, accessed, compared, reused easily. That standardized data is what lets AI tools actually learn the links between microbiome features and plant outcomes.
Okay, so the infrastructure is being built to handle this complexity too. Really reinforces that this isn't just about one patient simulation. It's about simulating and engineering entire biological systems,
Right.
bio-based planet almost.
That seems to be the direction, yeah. The ambition is huge. Okay, so we've laid out these three interconnected pillars, right? The AI reasoning engine like DeepThink, the consistent patient model with VSPs and CureFun, and the accelerated SynBio treatment design using tools like RF diffusion. The whole theoretical setup for personalized parallel simulation, it feels like the pieces are there.
The pieces are there in theory, but theory hitting reality, that's where the friction happens. We have these amazing AI tools, the SynBio techniques. What's the biggest single thing holding this back from really scaling up, from becoming mainstream today?
It's pretty clear across the sources. The number one roadblock, the most fundamental challenge, is data. Specifically, the lack of enough high-quality, well-labeled data.
The data bottleneck. We keep coming back to data with AI.
We do, because these deep learning models, especially the generative ones like RF diffusion that we need for designing new molecules, they are incredibly data hungry. They need tons of examples to learn from.
And SynBio just doesn't have enough of the right kind of data yet.
Not nearly enough, and often not in the right format. Across all the different areas, designing proteins, engineering microbes, understanding patient responses, there's a real shortage of adequately labeled, standardized data that AI can readily use.
So even the smartest AI, like DeepThink, can't work its magic if the information you feed it is messy, incomplete, or just missing.
Exactly. Its potential in life sciences gets throttled if the input is poor. You see this bottleneck everywhere. For example, getting comprehensive data that maps a specific chemical structure to how a cell actually behaves, its phenotype, that data is often patchy or locked away.
And without that link structure to function, you can't reliably train an AI to design new structures with desired functions.
Right. It slows down everything. Drug discovery, identifying active compounds, building accurate simulations. It all gets bogged down by this data deficit.
Okay, so data is the big hurdle. What's being done about it? How are researchers trying to clear that hurdle?
They're tackling it from two main angles, according to the sources. First, using clever techniques to make the most of the data they do have.
Like what kind of techniques?
Things like transfer learning. That's where you train a model on one task or data set where you have decent data, and then you adapt that learned knowledge to a different related task where data is scarce.
Okay, leveraging existing knowledge.
And also data augmentation, using mathematical tricks to artificially expand your existing data set, create more variations for the AI to learn from.
So technical workarounds, what's the other angle? Addressing the root problem.
Exactly. Formal standardization initiatives. Getting the whole community to agree on how data should be structured, labeled, and shared so that future data is clean, reusable, and AI ready from the start.
Mandating standards. That sounds crucial, but probably difficult to implement across labs worldwide.
It is difficult, but essential for building trust and enabling large-scale AI. The sources highlight some key examples. One big one is SBO, the Synthetic Biology Open Language.
SBL, what does that standardize?
It's a common language, a format for describing engineered biological parts and designs, how they're constructed, what they're supposed to do. It ensures everyone's speaking the same language when sharing designs.
Okay, standardizing the design information. What about standardizing the patient data or the experimental outcome data?
Yeah, other initiatives target that. There's NIPM, which focuses on niche phenotype mapping. That's about capturing data on how cells behave differently depending on their specific location within a tissue, like from histology images. Important for understanding disease and context.
Right. Location matters in biology.
And then there's GWS Central. That's a big repository for data from genome-wide association studies, studies that link genetic variations to specific traits or diseases across populations.
So gathering the genetic links to disease, which would feed into making those VSPs more realistic.
Exactly. It helps connect genetic blueprints to potential outcomes, providing crucial parameters for personalized simulations.
Okay, so standardization is step one for better data. But there's another issue with these powerful AIs, isn't there? The black box problem. If DeepThink designed some miracle molecule for a VSP, how do we know why it chose that design? We need to trust it before using it on a real person.
That's the critical challenge of reproducibility and traceability. And it's a major headache for AI and machine learning in general, but especially in medicine where the stakes are so high.
You can't just accept the AI's answer without knowing how it got there.
Absolutely not. The field knows complex AI designs won't be trusted or approved by regulators without a clear audit trail. So there's a big push to develop strict standards for data handling and, crucially, to implement provenance models.
Provenance models, like tracking the origin and history of everything.
Essentially, yes. A provenance model tracks the entire lineage. Where did the input data come from? How was it processed? Which version of the AI algorithm was used? What were the key intermediate steps or decisions the AI made?
So you can trace back the entire decision-making process.
That's the goal. We need that traceability to ensure the solutions aren't just effective in the simulation, but also robust, understandable, and ultimately safe for real-world human application. Without that trust and transparency, the gap between the simulation and the clinic is just too wide to jump. #TechOutro.
Wow. Okay, this has been a really deep dive into what it takes to build this future of personalized parallel simulation. Let's try to pull it together.
Yeah, synthesizing it.
We've got the AI brain, the advanced reasoning shown by DeepThink at the IMO, capable of handling that parallel complexity.
Check.
We've got the patient model, the VSP, using the CureFund framework, giving us consistent, high-fidelity simulation and even evaluation capabilities.
Got that piece too.
And then we have the accelerated treatment factory Symbio, combined with AI tools like RF diffusion and deep docking, designing totally novel, customized molecules incredibly fast with pathways towards production.
Right, the design and build component.
It's really the way these three pillars depend on each other that's so powerful.
That interdependence is key. This convergence, this blend of computational power and biological engineering. It really does feel like one of the most promising paths we have to tackle some massive global problems.
Beyond just individual health.
Oh, yeah. Think bigger. Climate change engineering microbes that eat CO2, food security designing more resilient crops or new ways to produce nutrients. This bio-industrial revolution, as some call it, has huge implications.
When you frame it like that, the potential scale of disruption is just immense. It makes you rethink what's possible.
It really does. And if you look at the most forward-thinking ideas in the source material, they're talking about solutions that sound like science fiction but might not be, like creating systems for on-demand, personalized food and medicine production for space agencies. Imagine astronauts on a long trip to Mars, essentially printing their specific nutritional needs or medications as required.
Wow. Okay, printing food and drugs in space.
Or localized production of advanced materials, or even bioelectricity, using engineered organisms right where you need it. The core idea is making highly customized biological solutions accessible, maybe even routine, for everyday life, everywhere.
That really brings it home.
So here's maybe a final thought, something for you to chew on based on all this. Think about something essential in your life today. Could be a medicine you take, a food you eat regularly, maybe even a material object you use.
Okay.
Could that specific thing be reliably, sustainably replaced by an on-demand, bioengineered solution within the next, say, 10 years? Because that's the level of transformation that these converging technologies are starting to make plausible. That's what you should mull on.
A bioengineered replacement for everyday essentials. That is definitely something to think about. We'll leave you with that provocative thought. We hope you enjoyed this deep dive. Keep exploring the science.
So keep questioning.
And we'll catch you next time.